Hey guys, welcome back. Today we're going to continue with what we've been talking about. We're going to move on from DNA replication to the next step in the process, which is DNA transcription and translation, and I'll talk about what the differences are here in a second. Sorry if it's a little darker than usual, it shouldn't be much of a problem, but I'm recording this a little bit later at night than I normally do. It shouldn't be a problem though. But anyway, today's going to be mostly notes because it's going to be similar to what we've been doing, but there's some key differences that I'll make sure you guys get. And I want to give you guys an opportunity to ask questions if you have them, especially now that we're on this distance learning. It might be a little confusing. I'm going to do the best that I can to explain it, but feel free to email me if you have any questions about this, because if this doesn't make sense today, the activity tomorrow is going to be based around this, so please ask any questions that you have. But in terms of like participation, the questions today are just going to be about the notes to make sure that you watched this video and that you read up and all that. So just make sure you ask any questions if you have them. Anyway, so up until this point, we've been talking about copying DNA, making more copies of DNA for more cells, because it's important that if you are going to divide and be a multicellular organism, or if you're going to reproduce asexually, the new cells have to have extra DNA. Why is the DNA so important? What is it for? Which we've talked about before. DNA is in charge of coding for all the proteins that make up your body, everything that you are, everything that you have in your body, around your body, everything that makes up who you are is controlled by your DNA. So this process of transcription and translation is actually taking the information in your DNA and turning it into the physical traits that you see. So the portion of your DNA that codes for eye color, this is what will contribute to that. So the actual instructions are in your DNA, but the actual pigment that gets formed is done through the process of transcription and translation. So the easiest analogy I can do to describe it is it's like getting information out of a library or like a recipe book. So you've got the recipe book. The book itself is just information. But if you take the information in the recipe book, copy it down, and follow the instructions, you can make a cake or cookies or whatever it may be. You'll make something out of it. So the DNA is the instructions. We're going to do processes to get the information out of the DNA and then turn that into actually something that we can use. So we're going to kind of break these up. I'm going to go over both processes today, and then we're going to focus on each one moving forward just to make sure we get it. We're going to focus on transcription first. We're going to do that tomorrow, but I'm going to introduce both of them today. So let's go ahead and get started. So two major processes in going from a DNA to a protein. Again, proteins being the things that make up your body. So you've got the DNA inside the nucleus. We need to get that information out of the nucleus, and then we need to translate that information into something usable. The information in a recipe book is useless if you don't know how to read. It will only give you the information that you need if you know how to decode the information in there. Sounds simple enough, but imagine if I gave you a recipe book in Spanish. For those of you, even if you are taking Spanish, I don't know how comfortable some of you are with Spanish. It would take a lot longer to decode because you aren't familiar with the language. So we have to translate that information into something that we can use. So two major processes. You have to go from DNA into something called mRNA, which I'll get into in a second. And then we're going to use that mRNA to make a protein. And that protein will vary in function depending on what it is that you need to do. It could be a protein involved in skin formation. It could be a protein involved in hormone regulation. It could be a protein involved in digestion, whatever it may be. There's lots of different proteins that do lots of different things. So each step in the process is given a specific term, transcription and translation. That's what we're going to do. This is called the central dogma of protein synthesis. Basically, what this means is this is the central idea of how proteins are formed. In order to form a protein, you have to go from DNA to mRNA. And in order to go from mRNA to protein, you have to translate what you transcribed. And you have to go in this order. It has to follow DNA to mRNA, mRNA to protein. So to make it simple, transcription is just the first step. DNA to mRNA, that's the first chunk. And then translation is the second step, going from mRNA to protein. So the way you can think about it is transcription. If you're transcribing something, you're copying it down. So the first step, we're just turning the DNA into mRNA. It'd be the same as like if you were given something in Spanish and you had to decode it in English. The sentences should line up and they should mean the same thing, but they're in different languages. Same idea here. You've got the DNA language, you've got the mRNA language, but we're going to use it. We need it as something usable, so we have to transcribe it into mRNA. Translation is actually taking that information that you wrote down and you say, okay, what did I write down? What does this mean? What have I copied? You're going to look at it, you're going to decode it, you're going to turn it into the protein that it's meant to form. So again, if you copy down a recipe from online and you write it down on your notebook, or if you write it down on your phone and you go back to make it, you're going to actually sit through it. You're going to go, okay, what do I have to do step by step? I need to do this, this, and this to make the protein. So first off, transcription. We're going to copy into something called mRNA. The M stands for messenger RNA. Again, this is basically just a different form of genetic information, but this way we can transport it. If you remember, DNA is found in the nucleus of the cell, and that's where it will always be unless the cell is undergoing mitosis. That being said, we can't really do anything with the information if it's stuck inside of the nucleus, so we need to have some kind of way to get that information out of the nucleus without moving the DNA. So that's what this is for. It's like a library. You've got all the information in the library, but if you're not allowed to take the book outside of the library, you're going to copy down the information before you leave, and then you can take your copy with you. Biggest difference that I know is going to throw off a lot of people, and I know is going to cause a lot of questions. In DNA, when we were doing replication, remember the 4 bases were A's, G's, T's, and C's. That is slightly different for transcription with mRNA for whatever reason. RNA molecules do not have the T. They do not have thymine. So anytime you're going to use a T in mRNA, you're going to replace it with a U. I'll talk more about that later when we get into practice tomorrow, and I'd like to bring up some stuff and show you some examples. But the easiest thing I can tell you for now, if it's confusing to you, the best thing I can tell you is, if it's easiest, just treat it like normal. Do it like you normally would with replication, where you're doing A's to T's, T's to U, or T's to A's, G's to C's, and all that. And then go back through and just switch all the T's for U's. I think that might be the easiest, least confusing way for some people. Some people might prefer just to do it right off the bat, but we'll talk more about it. But anyway, to recap, T's are going to become U's. So anytime you're going to see what would be a T pop up in the mRNA code, you're going to replace it with a U. So anyway, so translation. We're going to turn the mRNA strand that we just coded for into a protein based on the information that we have there. So if you remember, this is going way back, so I don't know if you remember, but each individual base pair along with the fragment that it's found on is called a nucleotide. So an A, a G, a T, or C is a nucleotide. mRNA is read 3 nucleotides at a time, so in 3 nucleotide chunks every single time. So you're going to read them 3 letters at a time. What this will do is that based on the combination, each of those will code for a specific part of a protein called an amino acid. There are 20 different amino acids that can be found in any living organism, and which amino acid gets placed into the protein will be determined by the sequence provided. So you can think of it like letters in an alphabet. You've only got 26 letters in the English alphabet. However, you can make millions of different words that all mean very, very different things. It's the same idea here. You've got 20 different amino acids, but there's a lot of different ways that you can get those 20 different amino acids. So here's how this works. This probably won't make much sense with the picture, but I do have a video that I'm going to link. Basically, what you've got here is the green strand is the mRNA sequence. So again, notice it's very similar to DNA. You've got your A's, G's, and C's, but now notice that instead of T's, there's going to be U's. So our first sequence is AUG. That's our first codon. And what's going to happen is that codon is going to code for a specific amino acid. That's the purple thing here. MET stands for methionine. I won't ask you to memorize that or know that. I'll always give you a chart. There's a whole chart with all the amino acids on it. You'll just have to plug and chug for which ones fit into the right place. So this first one codes for methionine. That'll be the first amino acid added. You can think of it like links in a chain. Each amino acid by itself is not a protein, but when you put them together, it will eventually make a protein. Once that's done, you just keep carrying on the process. So you go from the first set of 3 to the next one, to the next one, to the next one. Keep going until eventually there will be what are called stop codons, which eventually cause the DNA to stop coding, and you'll finish up your protein, and it will eventually form into the protein that it's meant to do, or is meant to be. So let me see. I know I'm skimming through this, but it's going to be, I'm trying to keep it as simple as possible for the distance learning. So if you want to go skim through this, you can. Don't worry a whole lot about tRNA for now if it's confusing. Normally we'd be able to talk about this in a classroom setting, but I really don't want to confuse you guys. So focus on mRNA. If these slides confuse you, skip them for now. It's not super important. Termination is just talking about how it ends. So you add links to the chain until you get to a stop codon, and then the stop codon doesn't code for anything, so then nothing gets plugged in, and it just lets go of the chain. The protein chain goes off into the cell, forms into the shape that it needs to be, and then it will do the desired function that whatever it's supposed to do. I will link this YouTube video showing the translation process. It's fairly short. I'm also going to link some other ones too, since again, this might be confusing and I want you guys to have plenty of resources, so feel free to look at any resources you need, and feel free to email me. Like I said, I would really prefer you guys email me if you have any questions, because I realize this is going to be confusing. Sorry, I need to silence my phone. This is for translation. We'll talk about this more next week. I just want to kind of show this real quick. And these are both the same thing, but I'll show you. This is what we'll use to determine what amino acids go where, and I know this seems really scary and confusing right now. I'll introduce it real quick, but if this freaks you out and you just want to focus on today's thing, you can cut the video off here because I really don't want to overwhelm you. But if you're curious, here's what translation will look like. I'll start with this one because this is my preferred chart. Some people don't like this one, but I think it's nice and easy because you just look for what the code is and then plug in whatever the amino acid is. So what you would do is if you've got your DNA sequence that you've coded into mRNA, and let's say, going back to our example, let's say the first sequence is AUG, and I want to say, okay, what does AUG code for? What's the amino acid? I just look at this chart, put on A, find AUG. AUG stands for methionine. I just plug in methionine. I'll go to the next one. Let's say I'm going to make something up off the top of my head, CUU. So let's go to CUU, leucine. Let's say it's AAG. AAG would be lysine. So all you have to do, I will always give this to you. I would never expect you to memorize all of these. I was never expected to memorize all of these, even in college. There's a whole lot of them, and they made this for a reason so people would have a reference so they didn't have to go back every time. So I will always, always, always give this to you. Don't ever worry about that. Like I said, I prefer this format because visually I think it works better, but I know a lot of people prefer this one. The way this one works is the first letter in the codon is the middle, so you work your way from the middle outwards, and then it'll eventually tell you what you need to code for. So for example, let's do the AUG example again. So A is the first letter, second letter is U, so that'd be down here, so we're narrowing it down to this fragment. G is right here, so we go AUG. The only thing that AUG codes for is methionine. Let's say it's CAU, C being the first letter, A. CAU goes down this one, that'd be histidine. Let's do GCC, that's original. GCC, alanine. Hopefully you get the idea. If that doesn't make sense, that's going to be some more next week stuff. We're going to spend all of next week probably working on translation because I really want to make sure we have it down. So to recap for now, just to make sure you guys understand today's portion, because I realize I kind of went over a lot, especially for distance learning. DNA to mRNA is transcription. We are transcribing it from one form of genetic information to another, and then we're taking that information, we're going to translate it. So DNA is the recipe. You're going to copy down the recipe by hand. That's your mRNA. Then you're going to take it home, you're going to read it, and you're going to decide, okay, what are the instructions saying, what do I need to make? And that's what translation is. You're going to turn it into a protein. So all I'm going to ask for today is just a couple questions about these notes, asking about what I just said about DNA transcription and translation, talking about the differences between mRNA and DNA, just to make sure you guys get the A's, U's, G's, and C's. That's really going to be it for today because I want to give you guys a chance to ask questions before we actually do some practice with this, because if this doesn't make sense now, I want to kind of get that out of the way before I get a bunch of people freaking out. Like I said, it's unfortunate that we have to do this through distance learning because this is definitely a section that I think most people would benefit from being face-to-face. But we'll do what we can with it. I will try to simplify it from what I've done in the past to make it as easy as possible. But anyway, answer the questions that I post. I will provide plenty of links and references as well. And then, like I said, please, please, please just email me if you have any questions about this. I realize I probably didn't go into detail as thoroughly as I could have, but again, we're kind of limited. And at this point, I'm just saying the same thing over and over again. So have a good day. Let me know if you have questions, and I will see you tomorrow.